Welcome everyone to Pathology Rounds. We've actually got a double billing today. We've got two speakers who are going to present on their research. And before I introduce them, I'll just maybe quickly do the land acknowledgement that I'm broadcasting from Vancouver, the ancestral and unceded territory of the Musqueam, Squamish, and Tsleil-Waututh nations. And I'd like you all to just take a moment to recognize where you're from and whose original territories those are. Our two speakers today, I'll introduce our first one, Martial Guillaud. He actually has a PhD in bioinformatics and biostatistics from the University of Joseph Fourier in France. But for a long time now, he's been a senior scientist here at BC Cancer, an adjunct professor of the UBC Department of Statistics, and an assistant professor in our UBC Pathology Department. His main research interest is in using quantitative image analysis, statistical machine algorithms, and 3D modeling of analysis of precancerous and cancerous lesions. So this is very topical in the current age we're in. And then our second speaker is Dr. Yasir Mohamud, who completed his doctoral training here at UBC in 2021. And he's now a postdoctoral fellow and early career researcher at the Center for Heart-Lung Innovation at St. Paul's Hospital. And he was actually a recipient of the inaugural CIHR Research Excellence, Diversity, and Independence Early Career Transition Award. And he's working to establish an independent research group, and his interests are in the intersection of virology, mitochondrial dysfunction, and viral heart disease. So I'll first let Dr. Guillaud start things off, and then we'll let Dr. Mohamud give his talk, and we'll have a little kind of time for discussion at the end. So take it away, Martial.
Thank you, Tyler. So today I'm gonna try to present you some of our work in that we call large-scale DNA organizations. I'll show you that it can be a promising imaging biomarker for epithelial cancer aggressiveness. So this is the three main points I will discuss today: an introduction on chromatin and cancer. I will describe our technology, the large-scale DNA organization, and show us some of our results on oral cancer, prostate, breast, and lung cancer. So in terms of chromatin generality, what we know about chromatin, and I know this audience pathologist, I will not teach you anything here, but we know that chromatin heterogeneity is a hallmark of pre- and cancerous lesion and is associated with cancer aggressiveness. It has been, of course, accepted that the physical structure of the nucleus is altered in tumor cells at the time of diagnosis. I can also say that there are several potential mechanisms through which changes in the physical topology of chromatid can broadly and non-specifically regulate gene expression. So also we know that in terms of, we show that histological analysis of a wide range of tumors often identify heterogeneity nuclear microstructure as a determinant for tumor formation and aggressiveness. And more often, and pathologists can seek and observe often that during tumor regenesis, there is some variation in clumping, size, and density distribution of chromatin within transformed cells. So here is a small cartoon about differences. Some of the differences you can observe between normal and abnormal cells, and we can see here the heterochromatin shows some changes. There is also an increase in nucleoli. The nuclear matrix is transformed and very changing in the overall shape of the cell due to the transformation of the nuclear lamina and nuclear envelope. So we can then observe these changes caused by the changes in the ratio and the proportion of chromatin and heterochromatin, which is then a change in gene expression and different modification, will impact some changes in the nuclear shape, in the DNA chromatin organization, and in the nuclear alteration. Here, example of, So here are just few examples. I know you know that better than me, again, that you can see some very difference in a normal bronchial cells, in a small cell lung carcinoma, in a large cell lung carcinoma, in the chromatin organization. Here we can see some PAP staining, and you can see, of course, some changes and modification in the clumping of the chromatin. Just few of these examples. Also, we know also that you can also see some changes here. In this case, here is in the transmission electron microscopy images of normal colonic cells from a normal colonic tissue in patient with adenoma on the left column, and in patient with an adenoma at the time of presentation. You can see here that even in a normal appearing cell, you can see some differences in the chromatin organization. So the goal of this large-scale DNA organization, basically what you're trying to do is to quantify precisely the changes that you see under your microscope in terms of nuclei shape and DNA chromatin organization. So for that, we're using Fulgen thionine staining. I'm sure you all know about this staining. This is a stoichiometric stain of the DNA. Basically, what you do in this briefly in this staining, and you first apply some acid hydrolysis to separate the two strand of DNA and to expose the purine aldehyde groups. We then apply the Fulgen stain, and the proportion of stain, the staining on this DNA is proportional of the amount of DNA in the nucleus. Here I'm showing the difference between the PAP stain and the Fulgen stain. As you can see here, we only stain the nucleus, and you can see on these images on the right some difference in the chromatin texture and the different clumping of the chromatin. Here, that was a study done to compare the stoichiometry of the Fulgen stains compared to the PAP stain taken at different image at different wavelength. And you can see that this is based on I believe, on cervical squamous epithelial cells, that the Fulgen cells give you a very narrow, sharp, normal peak of diploid cells. So that shows really the stoichiometry of this stain, that you really can quantify the DNA amount. This is based on cytology. On histology, you cannot assess the ploidy because this is a sectioning, so the cells are cut. But we know that the DNA is proportional to the Fulgen stain is proportional to the DNA. So obviously, I will not go into details, but we used to use some absorbance microscopy imaging system with high resolution camera. Now we're using different type of scanner. And you can see on the right an image of this type of nucleus under the CCD camera. On the bottom, I show here some gallery of normal diploid cells, cervical cells. On the right, some abnormal cervical cells. You can see, of course, again, it's no surprise for you, but increase in the cell area, in the nucleus area, sorry, in the boundary of the nuclei, and also, of course, in the nuclei, in the DNA chromatin organization. So here I can show you that what very important aspect of this work is to be very sure that we have a very sharp in focus nuclei. You can see here we can use some algorithm approach with the camera to identify which focal plane is the best to give the sharpest image here. We can also, the resolution is very important. In this case, we have some pixel of 0.30 micron. That gives the best resolution to extract your features. And here you can see that this resolution will also have a big influence of the precision and the accuracy of our features. Very important point in the last here, this is what we used to do in our lab in like, let's say 5-6 years ago, doing some classical image processing analysis to segment the nuclei. And you can see here, I'm going to show you this was an old method of doing things. And you can see here in the middle that it was very difficult to segment the nuclei before, but you could do some work here. I'm going to go back and forth, but it was still not perfect. And we know it was a big issue about this work is to have as many nuclei as possible with a good representation. So what we did, and this is the work of my colleague, Dr. Macaulay in the lab, we use now some kind of deep learning segmentation approach based on 2 different steps using the SUNet network. First, we do some center detection of the nuclei on the top right, followed by segmentation of this nuclei. And I want to show you some example here using this segmentation. This is a very complex scene of a lung cancer. You can see that there is, of course, a lot of overlapping. This section is about 4 micron thickness. And now I'm going to show you what we obtain with this deep learning segmentation. You can see that there is quite very good segmentation of nuclei. Here, in this case, I'm going to show you also, I don't know if you can see, but among 220 nuclei, you got maybe 10% errors, based when you compare that to some segmentation made by a human observer. So it's quite very good. And this new approach really changed drastically our results, because we now have a lot of nuclei we can use for segmentation and training. So once you collect this object or this nuclei, what we do, we extract some what we call LDO feature, large-scale DNA organization feature. There are different group of feature describing the morphology, the boundary of the nuclei, the densitometry, and the chromatin organization. So I will not go in detail here, but if you're interested, I can show you some, we can talk after, but basically the size feature describe the area, the perimeter. We have share feature describing the sphericity, the compactness, the elongation, using different type of image processing algorithm and to measuring the Fourier transform coefficient of each object. Also, we had very important feature here that are used a lot to discriminate different type of cell type, like lymphocytes, stroma cells, epithelial cells. This is called the nuclear boundary features. There's almost 30 of them. So that helps us to describe the shape of this nuclei, the boundary. Of course, we have all the density distribution features that are based on the Guillaud equation, that I mean the intensity of the light going through an object depends on the darkness of the object. I mean the amount of the quantity of DNA for each of these pixels. So we extract different features, the variance intensity, the optical density skewness, etc. But most important for what we're doing are the texture feature, and some of them are, for instance, the fractal texture feature that allow you to measure the spatial organization of the chromatin within the nucleus. There is other type of feature that we call the Markovian texture feature. I will not go in details, but it's a different way of looking at the pixel information within the nucleus. Some of these are other type of feature we call the rent length texture features. Here, I just want to show you more precisely. On the bottom right, you can see the different type of chromatin organization, the different type of chromatin, the low intensity, medium intensity, and high density chromatin. So basically here I show an histogram of a distribution of a schematic nucleus on the left. And we're gonna here measure what we call the discrete texture feature that allow you first to identify in the nucleus where are the pixels with low optical density, the ones with medium optical density, and the one with high optical density. And once you have this chromatin organized in 3 discrete regions, you can extract some features such as the area occupied by the high DNA intensity pixel, the low and the medium, the intensity of these different regions, the compactness of these 3 different regions, the location, and the different ratio of high, low, high, medium, etc. So you can extract a lot of different features like that. So I will not go into great details in terms of what kind of tool we're using. Once you have the object, to separate the junk from the good object, to have to separate the good cells from the bad cells. There is a lot of different algorithms we're using in terms of machine learning, such as the binary tree. We're using that maybe a few years ago. Now it has been replaced by machine learning algorithms such as random forest, which is a very classical that's used everywhere now. It works very well for us. So basically, I allow you to really have a very good classification of different group of cells. And in terms of the way we're doing our work here, and there is some variation, but basically we always try to separate cells coming from bad outcome patients from cells coming from good outcome patients. At the cell level, we end up with group of more than almost 100,000 cells from each of these different groups. Thanks to that, using some random forest algorithm, we can calculate some cell what we call a cell LDO score. From this score, we then come up with a classification, a binary classification. We have to move up to the classification, either the region of interest, if there is more than one per patient, or the core, if it's a TMA per patient. So we end up with a ROI or a core score. Then we transform that into a binary classification. And finally, to classify the patient into a good or bad outcome, we also use some kind of random forest or discriminant analysis or any type of modeling to come up with a score. And again, after we want to have a binary classification or good, bad, or even good, bad outcome or intermediate outcome. So this kind of algorithm is kind of a classical, and we use that for different type of work projects. So here, what I want to do here is to show you some of the results we got using this technology. And first, we're going to start with oral cancer. This is a work done with Dr. Liu and Catherine Poe from BC Cancer. And the goal of this subject was to try to use LDO to predict the nodal disease of oral squamous cell carcinoma. We used 35 patients. Here you can see the table. There was no difference in age, sex, and smoking history. And we defined then two groups of patients, one with that of a lymph node disease and the other one that didn't have any. So we had 16 LN negative and 19 LN positive patients. In total, for each patient, we have multiple regions of interest. So we have a total of 561 patients. So this is a schematic from the publication. So we did some imaging. As I explained before, there was some image segmentation, object classification of different type of objects, extraction of the feature, LDO features, and then after we did some kind of analysis. So the cell LDS score was used first to discriminate between cells coming from LN negative or LN positive patients. And here on the right, you can see the distribution of this score between LN negative patient and LN positive. You can see a big difference, and we go from a monomodal distribution to bimodal. So that could maybe suggest that this population on the right maybe represent an abnormal clone. So when you use this classification of cell in terms of using only grade 1 and 2 as a training set, we got almost 100% AUC for the classification of cells. Using the test cell, we got 90% of classification, correct classification. And using this time the region of interest, we got 95%. So when we use after, we transform that into a LDO score for the patients using the highest core LDO score. We use, for the training, we had 100% of sensitivity here and 92% of specificity. For the test, but will not, it's very good, it's 100%, there's only 6 cases. I will not, but the result on the training is quite good. In terms of ROC curve, you can see here we get 97% at the patient level, and here are the survival curve and for the training and for the test. So we got very good results here. Again, it was a small sample set, but we were very careful about choosing a training and test set. So this is the work we've done on breast cancer a few years ago. And the goal here is to try to, we had some TMA coming from UBC, and we wanted to see if we can identify using LDO the patient who survived, the patient who died from breast cancer. So we have 60 survivors and 35 deceased patients. So we use kind of the fact that on the TMA we had some 2 spots for each patient to split these 2 spots into training and test. So we use the same paradigm to try to do cell nuclei score, core score, and then patient score. So in terms of result cell LDO score, we had an accuracy of 76% with quite a good sensitivity and specificity at the cell level to discriminate these 2 groups of nuclei. Here, we didn't use random forest. You use linear discriminant analysis, and just looking at the time, I have to go faster. And here we use, this is the 6 feature that was found the most discriminant. They are mostly DNA chromatin features. In terms of LDO score, we had also a very good discrimination between the the bad outcome, the LDO positive and LDO negative cells. So I'm just going to go to the patient's result here. In terms of patient classification using LDO, we had a specificity of 99%, a sensitivity of 88% to discriminate these two groups of patients. And here you see the Kaplan-Meier curve between LDO negative and positive patient for the grade 1, 2 patient and grade 3 patient. So this is show that it's independent of the grade. And here we use a multivariate Cox regression model. And you can see in this case that LDO was the only, the most significant feature to explain survival and death of the patient compared to other classical features like grade, size, and node and age. So this confirmed the potential of LDO to predict the outcome of the patient. Now I want to show you some work done with prostate cancer. We had two projects with Dr. Keys and Dr. Macaulay. First was to try to predict prostate cancer response to brachytherapy using LDO. We had 106 patients. And here was the three group of patients that we classify into three groups. A group of patients that had no recurrence, 72 of them. A group of patients with early recurrence, so within 3 years, there was 19 of them. And a late recurrence group of patients that recur after 3 years. So you use these three groups, the group blue and orange here, no, group blue and green as a training, and we test on the late recurrence patients. So for the training sex, we use this group, the section from this good outcome and bad outcome patient, and we tested that on other type of section from these two groups, and the validation the set was consisted on 41 sections from the recurrence after 3 years patient. So here we had different type. We also run some random forest to try to discriminate using this kind of different. This is what we're doing now thanks to deep learning and algorithm, is to try to discriminate different type of nuclei using LDO feature like the stroma cell, epithelial cells, and immune cells, and we can after display them on the biopsy. Oh, by the way, here we're not using TMA, it was biopsy. So here, cell nuclei classification, we classify cell versus junk. And after we use the proportion of poor outcome nuclei divided by good outcome nuclei, to use that as a threshold and to try to discriminate these two groups of patients with good and bad outcome. At the training set, we had 100% of correct classification. The test set was 100% of correct classification, and the validation set was 94%. And here we can show the different rock curve. So we got very good result, and we hope now to be able to get some external group of patients from maybe from Canada. The second I want to show you here is the work done with Dr. Keys and Dr. Perkle. Here was to try to assess, to see if LDO could assess the risk of biochemical recurrence after radical prostatectomy. So we had a group of patients who exhibit some recurrence, 27 of them, and a group of 88 patients that didn't show any recurrence. So this is a typical what we did with the images showing the different type of algorithm. So in terms of LDO score, we can see here that the difference between non-recurrence and recurrence of different type of nuclei. And here was the confusion matrix based on the biopsy between no biochemical recurrence and BCR. You can see here that the specificity was 95% and the sensitivity is 70%. In terms of patient prediction, if we use 50% of the number of biopsies that you need to call a patient positive, we had also very good result here with a specificity of 96% and sensitivity of 85%. Here are the Kaplan-Meier curve between these two groups. And finally, I wanted to show you some results we got recently using 9-TMA of normal, early, and late stage adeno and squamous lung cancer using a segmentation, the new segmentation based on deep learning, when 279 patients. So we had group 1, 2, and 3 patients. Group 1 patient died within 2 years after diagnosis. Group 2 patient died within between 2 and 5 years. And group 3 patients are still alive after 5 years. So again, we use the group 1 as the poor outcome, the group 3 as good outcome, and we tested that on group 2 of intermediate outcome patients. So we'll go quite deep because I think I don't have too much time. We selected different type of 400 nuclei for each group, calculated the proportion of good and bad cells from different group of patients, always trying to be sure that we do training tested on different type of cells and patients. And here to show you that using that with done unsupervised clustering, you can now extract different type of cells based on this phenotype, this LDO feature. Like we had some cluster. So again, it was unsupervised cluster of epithelial tumor nuclei, some small roundish nuclei, some elongated nuclei. And you can see where these different type of cluster are in the spot, in the TMS spot. And you can see that some really clearly are in the gland and some are in the stroma. So it works. And using that, you can after see the percentage, how these different clusters, how they correlate with different clinical variables such as stage, tumor size, and eGFR here. And here, I just want to show you the result on that. So basically, we try to extract these to differentiate these two groups of good and bad outcome patients using this LDO feature. And we did a good job in terms of separating these two. So we use this is group 1 and 3 so it was a training set. So when you apply this algorithm onto the intermediate outcome, we were happy to see that this outcome only see the average survival time of this patient fall between these two groups. So it was very promising using only 5 features here. So I just want to finish clearly, but I hope I show you that LDO can be a robust marker epithelial cancer aggressiveness. We found very similar result in cervix, in Barrett esophagus, and in other projects in breast cancer. So the new AI-based algorithm likely gonna improve the performance of this algorithm. I also want to show you that we've shown in the lab that this LDO feature can also be a prognostic marker to predict progression to cancer in pre-malignant lesion in the cervix, in oral, and in lung cancer. It has been published, this result. And also, this is something we're trying to spend more time on these days, is to show, and we've seen that, but we're not sure yet that this can be applied in clinic, is that this LDO feature seems to be sensitive enough to measure changes in normal appearing cells in the vicinity of malignant cells. So he opened a new area of this research. And I think I'm done here. If you have a...
Thank you, Martial. That's very interesting, and I don't think I've followed everything in terms of some of those mathematical
Yeah.
methodological things, but it's certainly obviously a very powerful tool that seems like something that could be used to kind of complement our existing tools that we have.
Yeah.
And I see there's a question from Spence. I'm actually, Spence, if you don't mind waiting till the end of the session, maybe what we'll do is we'll let our second speaker, Dr. Mohamud, talk, and then maybe at the end we can do questions. So for those of you that missed my initial introduction, Dr. Yasir Mohamud is a postdoc fellow and early career researcher here at the Center for Heart and Lung Research at St. Paul's Hospital. And he's actually the recipient of the inaugural CIHR REDI Early Career Transition Award. And his interest is in the intersection of virology, mitochondrial dysfunction, and viral heart disease. So Yasir, are you able to share your screen to load up your slides?
Sorry, I'm having a bit of a technical difficulty.
Okay.
Just trying to connect. It seems to be saying that it doesn't have permission.
Okay, hold on. Let's do this. Let me help multiple here. How about that? Try it now.
Oh, not yet. Let me
Not
try
yet.
one more. it's asking me to quit and reopen. So maybe I can you can proceed with the question and I'll rejoin.
Okay, that sounds good. Thanks, Yasir. Sorry about the difficulties. Spence, are you there? Are you able to ask your question to?
Yeah, sure. Martial, thanks a
Hey,
lot for the
Spencer.
talk. Hey, I was wondering if you've gone back and looked at if there's any molecular changes that associate with the morphology or like the nuclear characteristics. Like if you looked at like translocations or p53 mutations or viral DNA.
No, we haven't, but yes, of course, we have to do that. But we haven't done that yet. But we can you hear me? But I believe if we want this technology to be used by pathologists like you guys, it's something we need to do to convince even further that we're measuring something that is something real. But we can show some correlation between some of the feature with some like EGFR positive different type of clinical classification of variable, but not at the degree I guess you're proposing here. I'm happy to try that. You're muted, Spencer.
Yeah, while we're still waiting, if there's any other questions, feel free to jump in. I also have a question too, but I don't. Okay, maybe I'll ask my question while we're waiting. But please feel free to either put your question in the chat or raise your hand. Martial, so this seems like it could apply to pretty much, oh good, yes, your slides are coming up. Maybe I'll hold off on my question just so we make sure we have time for Yasir's talk. Yasir, are you good to go ahead? I just don't hear, we see your slides, we just don't hear you yet. Maybe check your audio. Yep.
Oh, I think, yeah, I was muted. Let me get back.
Yeah, we can hear you now. And your slides were up there. If you just go into presentation mode, it should work.
Oh, perfect. Thank you so much.
Yeah, perfect.
Apologies for the technical difficulties. I'll try to speed up so we can get to the question part. Thank you so much, Tyler, for introducing me. My name is Yasir. I'm a postdoctoral fellow here at the Center for Heart-Lung Innovation. And for today's brief presentation, the title of this presentation will be Viruses at the Heart of Mitochondrial Dysfunction and Heart Failure. So I just wanted to give a brief outline of the presentation for today. I'll just initially address some of the mechanisms that we're uncovering at the viral heart mitochondrial interface. And I'll briefly touch on our ongoing project to study the role of mitochondria in inflammatory heart disease, leveraging some of the clinical samples that we have here at St. Paul's Hospital, and then just share a few comments on what we think the direction of viral therapy with respect to heart disease will be. So I just wanted to give a brief background. I realized that our department is quite large, and it's actually my first time giving this pathology rounds presentation. So I completed my PhD in 2021 at St. Paul's Hospital, UBC, and I'm currently completing my postdoctoral fellowship. And I'm fortunate to be funded by the Michael Smith Health Research BC Award, which is co-funded by St. Paul's Foundation, as well as the Center for Heart-Lung Innovation. And as Tyler mentioned, I was the recent recipient of the CIHR Research Excellence, Diversity, and Independence Early Career Transition Award. And I'm hoping to use the support of this award to transition to an independent research program and hopefully establish my own lab in the near future. So our research is focused on heart failure, particularly viral heart failure. And heart failure is a growing problem here in Canada. There's approximately 100,000 new cases each year. And surprisingly, some of the patients that we are seeing are patients like Paul here, who are quite young, active, and may not have a history of cardiovascular disease. And we believe this is a growing problem for the three main reasons, one of which is our aging population. But more interesting is that younger patients such as Paul are also showing up to the clinics with symptoms of heart failure. And we don't really understand why. We believe one of the reasons is this recent global viral pandemic, the COVID-19, that could have effect on viral heart failure cases. And my research group is actually interested in understanding how viruses such as not only enteroviruses, which I will discuss in depth, but other viruses such as emerging viruses can have an impact on our heart health. So we study in our lab a virus called enterovirus. They're a diverse family of viruses, mainly because they're quite common. They're responsible for nearly 1 billion global infections each year, and they have a diverse tropism. They can infect many different organ systems, from the central nervous system, the heart, pancreas, just to name a few. And they're associated with a diverse array of symptoms that range in severity from the common cold and mild flu-like symptoms to more serious conditions such as viral myocarditis and dilated cardiomyopathy. We study a cardiotropic virus called Coxsackie virus B3, or CVB3, that is associated with the DCM, dilated cardiomyopathy. And this is a condition where the heart cells in the ventricle are infected and injured, resulting in loss of contractile function. And what ultimately happens is the heart actually increases in size, but loses its ability to pump blood to the rest of the body, ultimately resulting in heart failure. And right now, the only viable treatments that give any success are heart transplants and replacing this damaged heart. So some of the challenges we face with viral heart failure is mainly twofold. On the one hand, we have knowledge that viruses such as CVB3 can directly infect cardiomyocytes. And in doing so, they're able to injure these cells and lead to loss of contractile function. And on the other hand, we find that there is an inflammation that is secondary to the infection. And this inflammation is intentioned to sort of respond to the viral infection. However, in certain circumstances, the inflammation is significantly pronounced. And instead of only clearing the virus, it can actually do damage to the heart. And both of these can combine in a pathological scenario, contributing to viral heart failure. So our body naturally has defense systems in place to help ward off the injuries that could take place following infection. And our first line of defense, as you may appreciate, are these physical barriers, such as the skin, mucous membranes, that help to block out viral pathogens, among many other different pathogens, from entering our system. But our group is actually interested more so on the second line of defense, following breach of the first line. And in particular, we're interested in the second line of defense because it's something that we have an incomplete understanding of, how viruses interact with our innate immune defenses. And expanding this understanding can help to expose potential areas where we can therapeutically intervene. As mentioned, the major challenge we face is not only heart injury from direct viral injury, but also immune activation and inflammation that contributes to the pathogenesis. So we're interested in the second line of defense. And this is, you can imagine, as the stage where the viruses come into direct contact with specific cell types. And with respect to viral heart failure, the cell type of interest is cardiomyocytes. And cardiomyocytes are these contractile cells that give the heart their ability to pump blood. And they're contracting approximately 60 to 100,000 beats per minute for the remainder of your life. And to accomplish this, they rely on these specialized organelles called mitochondria. So mitochondria are these double membrane structures, and they exist in all cell types, but they're most abundant in these high energy demanding cells, such as cardiomyocytes. They make up approximately 30% of the volume of cardiomyocytes, and they have diverse and essential roles. Not only energy production, which is very important for these high energy demanding cell types, but they play important roles in innate immunity as well. As you can appreciate here, mitochondria are very dynamic. They're constantly undergoing processes of fusion and fission. And beyond their role in energy production, they actually play a central role in our innate immune defenses. For one, they are a source of reactive oxygen species, or ROS, that can help to target specific pathogens. Additionally, they express key molecules such as the mitochondria antiviral signaling molecule, or MAVS. This is a protein that's localized on the mitochondrial surface that helps to signal antiviral defenses, particularly through interferon pathways, for example. Additionally, these mitochondria have their own genomic DNA, or mtDNA as it's called. And recent evidence is showing that mislocalization of mitochondrial DNA into the cytosolic compartment could be a pathological marker for several autoimmune diseases. And this process relies on these DNA sensors in the cytosol, for example, the cGAS-STING pathway. And most importantly, mitochondria play an important role in the cell life-death pathway, as injured mitochondria can release molecules such as cytochrome C, contributing to programmed cell death or apoptosis. So for the first part of my presentation, I'll mainly focus on how viruses directly injure cardiomyocytes. And we believe mitochondria are playing a role in this particular aspect of the disease. As you can appreciate, we previously, our research has shown that upon infection, in this case, not only cells, but even animal models, there is significant disruption of the mitochondrial network, significant collapse. Additionally, there's changes in the ultrastructure of the mitochondria within hearts. And I want to highlight some of the work that we are doing with the help of two very talented co-op students, Carly Lin and Wendy Huang. We sought to answer the question of understanding the mechanism of CVB3-induced mitochondrial dysfunction. And here we found twofold. On the one hand, we found that the viruses can significantly damage the mitochondrial membrane structures. And here I've stained, as previously mentioned, mitochondria are these double membrane structures. So they have an outer membrane as well as an inner membrane. And on this stain, you can see here in red, the mitochondrial outer membrane protein TOM20. And there's a stain here in green that's staining for the inner mitochondrial membrane protein, PISD, or phosphatidylserine decarboxylase. And the stark difference between control and infected cells is that upon infection, these inner mitochondrial membrane proteins mislocalize from their location within the mitochondria to the cell periphery. And mechanistically, they identified that expression of key viral proteins, and these are viral membrane-associated proteins, in particular, the viral membrane protein 2BC and 3A3D, can recapitulate individually when expressed in cells the phenotype that we observe following infection. So you can appreciate that in 2BC expressing cells, as well as 3A and 3D expressing cells, there's significant loss of the mitochondrial network compared to control cells. And interestingly, this is tied very closely with mitochondria-mediated cell death. So as I've clearly shown in the evidence prior, that viruses can in fact damage the mitochondria. And interestingly, our previous research has shown that a specific mitochondria recycling pathway called mitophagy is potentially impaired. So mitophagy is a process in cells that helps to recycle damaged and injured mitochondria through a process of autophagic clearance. And mitophagy plays an important role in many diseases, including the central nervous system, as well as heart diseases. And in the research that I've been conducting in my PhD studies, we found that these viruses in particular, enteroviruses, can significantly damage the autophagy process. So the major takeaway from this is that autophagy dysfunction actually impacts many diseases beyond just cardiovascular and infectious diseases. It touches on cancer, neurodegeneration, and even dysfunction becomes dysfunctional in the aging process. But our research focus is mainly interested in identifying the nexus of these three major arms, which is autophagy dysfunction, heart disease, as well as infectious diseases. And we think that mitochondria may be playing a role in all of these three facets. So just to summarize this first part of my presentation, our research is showing that CVB3, among other cardiotropic viruses, through expression of unique viral membrane proteins, can damage mitochondria. And as previously we've shown, these viruses can also disrupt an important quality control process called autophagy. And the result of this is that there's accumulation of these damaged organelles, mitochondria, that can impact viral heart disease. So for the second part of this talk, I'll quickly touch on some of the research progress we're making studying the inflammatory aspects of the disease. Mitochondria have emerged as there's this hypothesis that they are these ancient protobacteria that have developed an endosymbiotic relationship with the eukaryotic cells. And there's been early studies that tried to link or understand the interactions between mitochondria and an ancient antibacterial defense system called the complement system. And our lab finds this particular interaction very interesting, and we recently wrote a review on this process that is currently in the submission process. But I want to touch a little bit on the complement system and how we think it's playing a role in viral heart disease. So just to give a brief introduction on the complement system, it's this pathway that's been characterized for many years. In the late 1800s, actually, it's been identified. And it has these three major arms, the classical arm, the MBL arm, as well as the alternative pathway. And it follows these series of enzymes that form a cascade. And they ultimately terminate on a complex here called the membrane attack complex, or its other name is C5b9. And this system works very well in antibacterial defense. As you can imagine, these membrane attack complexes, or MACs, can form on the surface of bacteria, helping them to lyse, and then also drawing inflammation, phagocytic cells, as well as antibodies for their proper clearance. However, this pathway has been linked to several autoimmune diseases, and if unregulated, it can cause significant harm to the host cells as well. And fortunately, there are specific complement regulator proteins that help to block the pathway from pathologically activating. Among these, the complement regulator protein CD59 is one that helps to prevent MAC formation on host cells. Additionally, there are complement regulators such as CD55 and complement factor H that help to block some of the upstream steps of this pathway. So using the unique samples we have available here at St. Paul's Hospital at the Center for Heart-Lung Innovation, we wanted to understand how potentially this pathway could be affecting viral heart failure. And we're fortunate enough to have cardiac samples from explanted hearts. And this was an initiative that was started back in 1982 by Dr. Bruce McManus, who established the Cardiovascular Biobank. And it has since grown to over 100,000 cardiovascular samples. We're interested in understanding some of these samples, how their connections with viral infections could be contributing to the heart failure. And typically these samples look something like this. There's a control donor heart that we use to compare to cases of viral heart failure. And as you can imagine, there's significant immune infiltration in a case of viral heart failure. And the major two takeaways that we found from these initial studies was twofold. On the one hand, we found, consistent with our studies earlier, there's significant damage to the mitochondria structures within the heart. And secondly, we found significant activation of the terminal complement pathway upon infection. And these samples are hearts that we've tested and shown that they are positive for the virus that we studied, Coxsackie virus B3. And recently we've expanded this to include several other RNA viruses and viruses, DNA viruses, just to understand how they may potentially be playing a role in viral heart failure. Using a tissue microarray, we stained for these terminal complement markers, such as the C5b9 MAC protein. And surprisingly, we found that all of the viruses that we've tested have shown significant activation of the terminal complement pathway compared to our control cells. We're still in the process of including more samples into this, but the data is promising that this pathway may be pathologically activated following a viral heart infection. Similarly, we found that there's a strong association between mitochondrial gene expression, which we used as a proxy for mitochondrial injury, and cardiac inflammation. Using an online publicly available database funded by the NIH, there's a genotype tissue expression database, and focusing on specifically heart tissue, we were able to segment cells that demonstrated low mitochondrial gene expression from cells that demonstrate high mitochondrial gene expression. And we found that in cells expressing low mitochondrial gene expression, there was significant cardiac inflammation. And interestingly, in other studies, we were able to show that mitochondria themselves could pathologically activate the innate immune pathway. Here we used specific cell types, for example, macrophages, and we were able to use purified mitochondria as a whole or individually in parts, such as mitochondrial proteins, or maybe even mitochondrial lipids purified from mitochondria. And in all cases, we were able to see robust activation from these mitochondrial components or as a whole on the macrophage activation. Mechanistically, we're showing that mitochondria purified can also directly activate a specific complement pathways. The pathways that we've tested, including the classical arm, can be directly activated by mitochondria. And these are in vitro reactions in which mitochondria are purified and incubated with just serum, which contains the complement factors. And as you can appreciate, there's significant activation of C1R processing. This is one of the upstream complement proteases, as well as deposition of complement protein 4 on the mitochondrial surface. Interestingly, as I previously mentioned, there are specific complement regulators that help to prevent pathological activation, and they are CD55 and CD59 or protectin. We found that these proteins are significantly downregulated in infected hearts. And this was both in our human samples and infected animals as well. And mechanistically, we think that viral proteinases may be playing a role in the downregulation of these complement protective factors. These are viral proteases that function to typically process the viral polyprotein, but may have functions in also processing host factors. And we found that these viral proteases can directly target complement regulatory factor CD55. So just to summarize this second part of the talk, we believe that viral infection of these heart muscles is doing twofold. Direct mitochondrial injury is contributing to the pathological activation of innate immune cells. At the same time, downregulation of these complement protective factors is contributing to the pathological activation of the complement pathway. And together, we believe these are converging on potentially describing a novel scenario by which complement factor is initiating viral heart injury. So this final part, I want to just summarize what we think is the next direction. As you can imagine, viral heart disease, there's no viable treatment options beyond heart transplant, of course, which has its own limitations. Beyond the limited supply, organ rejection is a very common issue. So we're looking to establish new therapeutic strategies that could address several important components, including demonstrating antiviral potential, being immunomodulating, and demonstrating some capacity for repair and regeneration of the injured hearts. And we believe mitochondria may be an important target as they impact these processes very intimately. And our next research goals are to try to identify key mitochondria targets that can be leveraged for this therapeutic initiative and develop a biologically relevant model to study these therapies and hopefully establish a therapeutic pipeline to test them and move them along to patients ultimately. So with that, I would like to conclude my presentation and acknowledge the wonderful colleagues that I have here in the lab. And I'm happy to answer any questions.
Wow. Thank you, Yasir and Marcel. Excellent talk, Yasir. For me personally, this was particularly fascinating because one of my best friends in high school, his younger brother was 19. He had a viral infection, got over that, and he was he would exercise in the gym every day, and he went back to the gym his first day back, and he tried to lift weights, and he collapsed. And he had end-stage heart failure. His ejection fraction was around 20%. He was transferred to St. Paul's Hospital, where he's on this cardiac ward with all these older people and just him, this 19-year-old. And he basically ended up getting a heart transplant. And they attributed it to this, exactly what you're saying, this post-myocarditis or post-viral myocarditis. And presumably it was something very similar to what you're describing. And he's fortunately alive today in his early 40s now. But basically it was only because of a heart transplant. Otherwise, he wouldn't have survived longer than a year. So obviously this is very important. And so I guess related to that, my question is, there just must be something that's, given how ubiquitous this virus is, or these viruses are, there must be something about certain individuals in terms of susceptibility. And do you have any sense of if you can use these people that have this unfortunate reaction to the virus, if you can identify certain genes or whatnot, or is there a sense of if you can use certain individuals to kind of drill down further on this?
That's a wonderful question, Tyler, and thank you for sharing that story about your friend. That's actually a I often don't hear it, but I've read about many stories similar. But to hear it from an actual person who's experienced it as well, it's quite like it makes sense, the research that we're doing, the importance of it. And on the second point about what we can identify, I think it's something that we're actually very keen on identifying, because these are conditions that don't occur on everyone. There's a specific group of individuals that are at more risk of developing viral heart failure. We don't really understand why. We think genetics obviously plays an important role, and of course the environment as well. So in order to have this pathological manifestation, there has to be the right environment, as well as the risk genes must be there. And one of our goals is to try to identify, leveraging some of the clinical samples we have here available, to identify key risk factors. Our work in the complement pathway is suggesting perhaps maybe variations in complement-mediated pathway reaction could be playing a role. We're hoping to look more deeply into that. But I think it's a future direction that we're really interested in and want to pursue.
That's great. Thanks, Yasir. Any questions from the general audience for either Martial or Yasir? Yasir, out of curiosity, have you engaged at all with Ed Conway? He's a big complement researcher out at UBC Center for Blood Research.
I actually have not had a chance, but this is because this is a really new field, actually. Just in the past few months, it's materialized. But I'm really glad to hear that we have people here at UBC. I hope to reach out to Ed, gain their expertise. We're quite new to it here. So Ed Conway is a name that I'm going to...
Good. Yeah, Ed, he's actually recently been exploring some areas around complement and metabolic syndrome as well. So he's definitely interested in complement and cardiovascular disease and any links there.
Wow, very interesting. There's actually the students that I highlighted, Carly and Wendy, they wrote a very nice review. And there's this dichotomy between like the extracellular complement and intracellular complement, or the complosome. I'm really keen to hear what he thinks about, because a lot of it is quite controversial. Like no one really understands what's happening with this pathway, but it's like an exciting direction.
Yeah. Well, I think we're at 12:57. So if there's no other questions, I'd like to thank both Martial and Yasir for really just highlighting two very different, but also equally interesting areas of research going on in our department. And I think this is a really good demonstration about how, we've got people in our department that are working in different silos, that it's really good to have these opportunities for these kind of sharing of knowledge, and potentially maybe even people might be able to reach out to you both if they see a potential area of collaboration. So thank you, Martial and Yasir, and hope everyone has a nice weekend, and despite the rain coming up.
Thank you, Tyler.
Thank you, Tyler.
A good day. Bye.
Good weekend. Bye now.